Today's topic for discussion is a topic of plant pathology, and today's disease for discussion is wilt disease of Cajanus cajan. In Marathi, the crop plant is known as tur plant. It is the best source of protein, and the wilt disease, it is a very severe and major disease of pigeon pea. It causes heavy losses of the crop, and in our country it is very common in Cajanus cajan growing states of India, especially Uttar Pradesh, Madhya Pradesh, Bihar, and several parts of Maharashtra and Karnataka. This disease is very severe. Let us see the symptoms of the disease. Symptom means the expression of the disease. After interaction of the pathogen and host, a specific symptoms are developed in the plant. Abnormal things occur in the plant. That expression is known as symptom. The susceptible plants mean those plants which can infect very easily. It has no resistance power. Such plants are called susceptible plants. They get attacked when they are about 5 to 6 weeks old. The symptoms are variable. What happens initially? The leaves becomes yellow, and later on the leaves fed up and most of the leaves fell off on the ground. This is defoliation. The plant is in a blighted condition. Entire plant, because of no leaves on the plant, that gradually dry up. And why it dry up? Because of the pathogen inside its xylem tissue, there is no supply of water to the plant. Therefore, the plant gradually dry up, and all the leaves initially they becomes yellow, and then after that they dry up and fell off on the ground. The leaves appear to be affected progressively from the bottom towards the top, and sudden wilting is very rare. The plant dries up gradually, step by step. Finally, the entire plant completely dries up, and in severe cases of infection, more than half of the plant in the field may dry up, and in severe cases and heavy losses of the crop is called. The wilting is brought about by plugging of the vascular tissue of the stem and roots of the host plant. What happens when the pathogen enters into the host through the root hairs, and gradually it moves to the xylem tissue, and xylem vessels are blocked. Xylem is the pipeline of the plant which helps in supply of water from soil to plant. The xylem tissue, this pipeline of the plant, get blocked by the growth of the fungus inside the xylem vessel, and because of this, because of excess growth of the pathogen inside the xylem tissue, the water supply to the upper side of the plant is blocked, and because of this the wilting occurs, drying of the plant takes place, and the fungal hyphae inside the xylem tissue, they produces a specific toxic substances which can kill the plant cells and concern in the ascent of sap. And in case of where plugging of the vascular tissue of the host takes place on one side only, the wilting may be partial. Sometimes partially wilted plants are also observed in the field. This is the details of the symptoms of the wilt disease of pigeon pea. Let us see all about the causal organism. Causal organism or pathogenic organism which causes wilt disease of pigeon pea. The name of pathogenic organism which causes wilt disease is Fusarium oxysporum from species Udum. This is the name of the pathogen, and this is the fungus that consists of septate hyphae. Hyphae are septate, and initially the fungus grow between the cells. That means it is intercellular in the initial stage, and later on the hyphae enters inside the host tissue. That stage of the hyphae, that is termed as intracellular. Later on it becomes intracellular. Okay. And it has also observed that after infection, the xylem tissue becomes reddish in color because of the pigmentation secreted by the interaction of the pathogen with the host cells. Okay. Let us see the disease cycle. In disease cycle, the fungus reproduces by asexual reproduction, and asexual reproduction takes place by formation of two types of conidia. 2 to 3 kinds of spores are produced, but conidia are produced which are of two types: microconidia, micro means very small sized conidia, and macroconidia, macro means large sized conidia. This fungus also produces chlamydospores, which are thick walled, and formation of chlamydospores takes place in mostly in adverse environmental conditions. Let us see the microconidia and macroconidia are produced on the hyphae, on the fertile hyphae, which are known as conidiophores, and the microconidia, these are generally very small, and they are produced in large number at the tip of conidiophore. Conidiophore means conidia producing stalk-like structure. At the tip, many microconidia are produced, which are indistinguishable from the vegetative hyphae. The spores may be rounded, curved, or elliptical. The microconidia are very small, somewhat elliptical in shape, and formed within the tissue of infected host. Usually the microconidia are unicellular. Uni means one-celled structures. Sometimes one or two septa are present in the conidium, in the microconidium. In cultures, when you will grow this fungus in the laboratory, in cultures they are held together in a ball or false head. Many conidial balls are formed in the cultures when you grow this fungus in the laboratory on media. Okay. Let us see how are the macroconidia which are produced by the Fusarium oxysporum fungus, which causes wilt disease of pigeon pea. Macro means large conidia. Macroconidia means large sized conidia produced by Fusarium oxysporum, which causes wilt disease. And this macroconidia are long, sickle shaped means somewhat curved, or crescent shaped structures. Okay, and they are septate and have pointed tapering ends. Macroconidia are having 3 to 4 septa. The macroconidia are also produced at the top of conidiophore. Conidiophores means conidia producing stalk-like structure. And at the top of conidiophore, conidia are also produced. And the whole fruit-like body is formed by the excess growth of the conidiophore which are closely associated on the infected part. Such a cushion-shaped stromatic mass of hypha is developed, that structure of the fungus that is known as sporodochium. Okay, and the macroconidia are released very soon, and whenever they reach to the proper host plant, they causes disease, or they remain in absence of the host plant, they remains on plant debris in the soil, because this is a saprophytic fungus and this is a facultative parasite. In absence of the proper host plant, that remains in soil on dead organic matter, and whenever it comes in contact with the proper host, it causes disease. Next, this fungus, it also produces another type of spores which are known as chlamydospores. The chlamydospores are thick-walled spores. These are drought resistant spores, and mostly they are round or oval in shape. And these spores are formed in the hyphae. Hyphae are septate In each septa, one chlamydospore is developed in adverse environmental conditions. They occur singly or they occur in the form of chain. And sometimes they are intercalary to the hypha, or they are formed at the terminal region of the hypha. The chlamydospores become separated from the parental hypha after maturity, and the chlamydospore independently can infect to the new host plant. But generally the chlamydospore, it is also known as resting spore. It can remain in soil or on dead organic matter for long duration. Okay, next. Let us see details about infection. In PPT, a photograph is shown. This is the longitudinal section of the infected stem. See the xylem tissue, which is dark red colored, shown in the photograph. That is because of the infection of Fusarium oxysporium to the stem of pigeon pea. Let us see the details of the infection. Wilt disease of pigeon pea, it is a soil-borne disease. Its spores remain in soil, and the microconidia and macroconidia and chlamydospores that remain viable in soil for long duration, and at the sowing time the spores germinate and initiate new infection. The germ tube is produced in presence of moisture, and infection takes place through the young roots. Young rootlets of the plant get infected by soil-borne fungal spores. Infection of aerial part of the plant takes place through airborne spores. The spores are very light in weight. They are easily blown in the air, and even the airborne spores, when they comes in contact with the host, they infect the plant. The fungus mycelium also exists in soil as an active facultative saprophyte so long, and dead host root also acts as primary source of primary infection, and other parts are also present in the soil. It gains entry into the host through rootlets. This is important. Through the rootlets, it reaches to the xylem vessels of the plant, and inside the xylem vessel it grows, and excess growth of this fungus inside the xylem tissue is the main reason of blockage of the xylem tissue, and water supply of water stops because of the growth of the fungus inside the xylem. Next, let us see the control measures of the disease. See that for the management of the disease, we have to use resistant varieties. Use resistant varieties for sowing purpose to avoid the disease. Second thing, field sanitation. Before sowing, entire field you have to clean, remove all plant debris from the field and burn it away from the field. This is the field sanitation. This can reduce the disease intensity. 100% management of this disease is difficult. Okay, crop rotation is one more method to manage the disease. In crop rotation, what happens? Some pathogens, they infect to a specific host plant, because of so that we can, if we rotate if we follow the crop rotation, we can manage the disease for some extent. Rotation of the crop is effective against the disease. And before sowing, you can treat the seeds. If this fungus also occurs on seed, on the surface of the seed, and that can be removed with the if we treat the seeds with recommended fungicide. And there are several fungicides. Recently new fungicides are available in market. Even eco-friendly biopesticides are also available in market. Use of recommended fungicide and with the recommended dose is effective against disease control.